Thank you again for that introduction, and it's a pleasure to be here today. I will be talking about my research that looks at the impact of the porcine reproductive and respiratory virus on amino acid protein metabolism as well as protein deposition. So in spite of extensive biosecurity measures that are implemented in modern pork production units, the industry is still faced with disease-causing challenges that highly impacts the sustainability and profitability of pork production. Upon exposure of the animals to antigens, there is an increase in the release of pro-inflammatory cytokines from innate immune cells such as macrophages. These pro-inflammatory cytokines can directly, through the receptors which are found on almost all tissues, and indirectly, through the hormonal and neurological response, shift the animal's metabolism as well as impact the physiology and behavior. This alteration in metabolism can highly impact the pig's productivity. So for example, in a study done by Williams et al. In 1997, he showed that when 10-kilogram pigs were exposed to a very mild immune system stimulation, it highly impacted the growth performance of the animal, inhibiting its ability to perform to its genetic capacity. So in short, disease is expensive. I've now introduced to you the concept that disease impacts the animal's performance, but I'd like to talk more specifically about how disease alters amino acid utilization, since that's where the focus of my research lies. So during the activation of the immune response, there is an increase in newly synthesized proteins and metabolites, such as acute phase proteins. These newly synthesized proteins or metabolites require additional needs for specific amino acids, which in turn can qualitatively and quantitatively impact amino acid requirements. The additional need for these specific amino acids can increase muscle protein degradation, as well as redirect nutrients that would normally go towards growth processes towards response of towards support of the immune response. Now, based on previous studies, sulfur amino acid, threonine, tryptophan, phenylalanine, leucine, isoleucine, valine, and glutamine putatively become more important during disease relative to other amino acids. However, a majority of these studies were conducted under various nutritional states. They use various disease models, with some being more severe than others. The animals were of various age and gender, and some would base their conclusions off of observed changes in metabolic demands, which we now know that a metabolic demand or a change in metabolic demand does not necessarily mean a change in dietary requirements. Additionally, some of these studies would only look at the impact of disease on one specific amino acid, which ignores the relationship among other amino acids. And finally, they use various models of measurement to look at the impact of disease on amino acid utilization. Some of these studies mentioned used a measure of concentration. They would look at a single measure of concentration in order to study the impact of disease on amino acids. But we now know that concentration can be maintained from amino acid influx from dietary protein and or protein breakdown, and amino acid efflux, which is going towards our amino acid catabolism and oxidation, or protein synthesis. So it's safe to say that using a single measure of amino acid concentration could potentially be misleading. Now, a better alternative to look at the impact of disease on amino acid utilization is to use plasma amino acid kinetics or plasma amino acid flux. Because even when concentration is constant, changes can still occur, reflecting the modification of amino acid utilization during different metabolic states, such as disease. Therefore, the objective of this research was to evaluate the effects of immune system stimulation induced by the PRRS virus on plasma amino acid kinetics as a measure of amino acid utilization, as well as apparent ileal digestibility of protein and whole body protein deposition. The general design of this study utilized 20 PRRS negative gilts that were individually housed in metabolism crates at a BSL-2 facility. After a 3-day adaptation period, the gilts were surgically catheterized in both the left and right jugular and then allowed to recover for 7 days. They were feed restricted at 550 grams per day, a corn, soybean meal-based diet. I must mention here that 24 hours prior to isotopic infusion, the feeding period increased every 4 hours in order to minimize amino acid diurnal pattern. An immune system stimulation was induced by intramuscular injection of a live-filled PRRS virus. For measurements, we took serial blood collection at time 0 and every 48 hours after immune system stimulation until the completion of the study. It was analyzed for hematology, blood chemistry, and viral load. Eye temperature was taken every day using the infrared imaging technique. We conducted 2 nitrogen balance periods. There was a 3-day pre-nitrogen balance period and a 3-day post-immune system stimulation nitrogen balance period. And I'd also like to take a note here that the 3-day post-nitrogen balance occurred 6 days after inoculation with the PRRS virus in order to allow for enough incubation time of the virus. And we also measured apparent ileal digestibility using the slaughter technique and titanium dioxide as an indigestible marker. I'd also like to mention that for this measurement, we had a control group of pigs that were injected with saline that were used as the reference group. At the end of each nitrogen balance period, each pig received a single dose of universally labeled C13 N15 amino acid mixture. Serial blood collections were taken in the times outlined on the slide and used to measure isotopic enrichment, and the plasma amino acid flux, i.e. the sum of disappearance from the amino acid pool, was measured. Basically, this is a two-pool model that measures the rate of disappearance of amino acids from the plasma general pool that go towards catabolism and or protein synthesis. So what I'd like you to take away from this side is that we used a double exponential model and fitted it with enrichment data that allowed us to get the parameters necessary in order to calculate each individual amino acid flux and pool size for each pig. So this brings me to my result. This top graph here represents the viral load of the PRRS challenged pigs. It was measured in genomic copy per microliter, which is represented on the y-axis, over time on the x-axis. And as you can see, the viral load began to increase only 48 hours after inoculation, and it reached its maximum viral load at 4 days post-inoculation. Disease was further validated by elevated eye temperature, which increased 48 hours after inoculation and remained consistent thereafter. This elevation in body temperature is a good representation of the body's natural defense against an infection. Before I get started with the blood chemistry results, I would like to set the stage for this graph as well as the ones that are to follow. The blue bars will represent the pre-ISS period or the pre-challenge period, and the red bars will represent the post-challenge or the post-ISS period. For the blood parameters, please take a note that each parameter was measured in its representative units, which are outlined next to the parameters on the x-axis, and the y-axis represents the numerical value for which it was measured. As you can see, relative to the pre-ISS period, there was no significant change in blood glucose, there was a significant increase in anion gap, there was a significant decrease in hemoglobin, no change in blood urea nitrogen, and there was elevated creatinine levels. Taken together, these blood parameters show the shift in metabolism that occurs during disease and is consistent with previous findings. For these two graphs, I'd first like to focus your attention to the graph on your right, which represents the apparent ileal digestibility of dietary protein. And as I mentioned before, we had a subgroup of pigs that were used as the comparison. And as you can see, the PRRS infection significantly decreased the apparent ileal digestibility of dietary protein which could most likely be attributed to the increase in gut endogenous amino acid losses during disease. Now to draw your attention to this graph here on the left, this represents the protein deposition, which was measured in grams per day and is outlined on the y-axis. And as you can see, relative to the pre-ISS period, PRRS infection significantly decreased the animal's ability for protein deposition. And both of these results are in agreement with previous findings. This brings me to my plasma amino acid flux results, which is a good indicator of amino acid utilization. On the x-axis, we have the amino acids that were measured, and on the y-axis represents the flux or the utilization of these amino acids in micromoles per kilogram per hour. Relative to the pre-ISS period, there was a tendency to decrease tryptophan utilization, suggesting that there is a decrease in the metabolic need for tryptophan during disease. But more importantly, we saw a significant increase in both methionine and threonine for its flux or utilization, suggesting that these amino acids more likely become the most important during disease situations. As far as the amino acid pool size, you can see that infection with the PRRS virus significantly increased or tended to increase the pool size, which is a good indicator of muscle protein breakdown. And this was also further validated by the increase in daily urinary nitrogen excretion and elevated creatinine levels. So in summary, immune system stimulation with PRRS virus reduced the apparent ileal digestibility of dietary protein as well as protein deposition. It increased the methionine and threonine flux and tended to decrease tryptophan flux. And it did not affect the utilization of other amino acids. So in conclusion, the increased utilization of methionine and threonine could most likely be attributed to the synthesis of immune system metabolites and or increased catabolism of these two amino acids. And the increased methionine and threonine utilization may impact other dietary requirements relative to other amino acids. However, further studies are needed in order to evaluate this implication. And amino acid kinetics provides a better assessment for amino acid utilization than a single measure of concentration.